Hello and welcome back. Today is all about DNA replication. This is sort of taking what we've learned in the last set of notes and applying it in a real world sort of way. This is what happens in all of your cells all of the time. It's really important to understand the process of DNA replication, and this in particular is like just scratching the surface. There's so much more involved, but this is all that you as a ninth grader, or at least in this biology class, need to remember and need to understand. So let's get started with a little scientific history. There were two competing, groups of scientists that were trying to determine the shape of DNA. And they worked at the same time, but in different locations, and they were kind of racing to get their information published so that they could, claim the fame that went along with determining the shape of DNA. Now keep in mind, this was in like the 50s. This was not that long ago. I know the 50s probably seem like a long time ago for you, but in the grand scheme of things, this is relatively recent. So prior to this, no one knew what DNA looked like, and no one really knew, specifically what was going on in there. So the two groups were Rosalind Franklin and Maurice Wilkins, which I have a picture of them on the next slide, but not on this slide. They were the first, officially the first, to study the double helix shape. And we know that DNA is shaped like a double helix, and they studied this using X-ray diffraction images. This image right here is an X-ray diffraction image, and this image almost always shows up on an SOL exam. So, just recognize what this is and who did it. And it is really because of Franklin and Wilkins that this image even exists. Well, in the spirit of collaboration, Franklin and Wilkins shared this image with Watson and Crick. And Watson and Crick took that and ran with it. And so Watson and Crick are actually more often than not given credit for discovering the double helix shape of DNA. They had that image and published their paper first. So they're given credit for discovering this. And when you research about DNA and its origins, you will almost always hear about Watson and Crick and not Franklin and Wilkins, which is a little upsetting. It's even more upsetting because in 1962, Watson, Crick, and Wilkins received a Nobel Prize for their work on discovering the structure of DNA. But Franklin had actually passed away 4 years before the Nobel Prize due to cancer, and Nobel Prizes are not awarded posthumously, so if you pass away, you cannot claim a Nobel Prize. So she's not even given credit on the Nobel Prize of this aspect of this discovery, which is a little bit frustrating, but here we are. When you are learning about this, you're going to hear a lot about Watson and Crick, but please remember the vital contribution from Maurice Wilkins and Rosalind Franklin. Really important. So now let's talk about the main purpose of DNA replication, which is to make one double helix into two identical, write that down, underline it, identical double helices. So you're going from 1 to 2. Now remember, all of this happens in a cell before the cell divides, right? Before the cell goes through mitosis. In interphase, during the S phase of interphase, the DNA replicates itself. So DNA replication happens in sort of 3 main steps. The first step is to untwist and unzip that double helix. Right now it's all twisted nice and tight, and we want to untwist that and actually separate it out. So we're going to break those hydrogen bonds between the nitrogen bases, and by breaking them, the way that we do that is using an enzyme. Oh yeah, enzymes, they don't go away. We're going to keep learning about different enzymes. Remember that an enzyme is a protein. So helicase, that's how you say that, is responsible for untwisting the DNA and also coming through and just ripping through these hydrogen bonds, breaking them open, and allowing these nitrogen bases to be exposed. That's the key. We want them to be exposed and not already connected to the other half of the DNA, okay? So first step, untwist and unzip because of helicase. The second step is a second enzyme, so DNA polymerase, yep, polymerase, that's how you say that, is like I said, another enzyme, and its job is to pair up bases using the same base pairing rules we already know, right? A to T, G to C. And there are free nucleotides kind of just like floating around in our bodies, in our cells, right? And so DNA polymerase gets those free nucleotides and lines them up appropriately. So depending on what the order of the bases are, DNA polymerase will find the complementary base pair and line it up to make a new strand of DNA. The third and final step is, like wrapping it all up, right? We're done. We have separated it. We've made two new identical strands of DNA, and now we're kind of like finishing up. So end results. Once all of those base pairs have been matched thanks to DNA polymerase, we now have two brand new DNA molecules, right? They are their own double helix, right? So this kind of like top piece with the blue and the red is identical to this bottom piece, the blue and the red. Those are identical to each other, and both of these are identical to the original strand, right? When you go through DNA replication, at no point are you changing the sequence. You are just making copy after copy after copy. It's always the same sequence. You're just doing it again and again and again so that you have more DNA. You're making more cells that have the same DNA. You picking this up, right? It's all identical, right? This is like very key. Please remember that you are making identical DNA molecules. Now let's talk about this last thing. This is your last slide for today. When we are going through DNA replication, the process is called semi-conservative. And maybe you've heard of semi before, right? Semi-permeable membrane, right? Some things can come in and out of the cell membrane. Well, semi-conservative means some things are conserved or kept and some things are new. And that is exactly what happens with DNA replication, because when you make a new DNA molecule, you are making a DNA molecule that has half of the original or old DNA strand, or you keep that, right? And half of it is brand new thanks to DNA polymerase. So looking at this image, right, this dark red is your parent strand. That's your original. When you go through replication, half of the new DNA that is created is the old parent DNA, right? That is the conserved part of DNA replication. And then this light pink, which is kind of hard to see on this slide, or maybe I'm just looking at it at a weird angle, but this light pink part, that's new DNA. So this is not from the original parent strand, but it is going to end up being identical to the parent strand. So now, semi-conservative, partially kept, right? So like you use half old, half new. Really important. We'll talk about this more in class, but right now, great job. I'll see you later.